Good morning. I am Eirini Papapetrou. I'm an associate professor here in Oncological Sciences in Sinai, and it's my great pleasure to welcome our first keynote speaker for this session, Chuck Murry. Chuck started his scientific career in North Dakota, graduating with a chemistry degree, and went on to complete an MD PhD program at Duke University. He trained in pathology at the University of Washington in Seattle with a focus on cardiovascular diseases, and he stayed at the University of Washington and is now, to this day, he's now a professor of pathology and also the director of the Institute for Stem Cells and Regenerative Medicine, also known as ICRM. Chuck's research focuses on deriving cardiomyocytes from human pluripotent stem cells, and in the past few years, this work has matured enough, and he'll tell us about it, to provide proof of principle in non-human primate models of its efficacy. And with these very promising, exciting results, setting up the stage for clinical trials aimed at replacing cardiomyocytes with cardiomyocytes damaged myocardial tissue in patients who have suffered myocardial infarctions. So Chuck, thank you so much for coming, and we look forward to your talk.
So good morning, everybody. And first, I'd like to thank Eirini and the rest of the organizers for the Sinai Innovations for the opportunity to address you this morning. It's really quite an honor, and only 50 years. I always thought this was an older and more, just given your distinction and level of accomplishments, I figured this was like an old-time school. And so anyway, congratulations on that, and here's to the next 50 years. What I'd like to do today is to tell you about the work we've been doing in the upper left corner of the United States, working on trying to regenerate the heart. This is a work in progress. If you were hoping that I was going to come in and tell you this is a done deal, this is not a done deal. Some things are working, we're struggling with some other things, and I'll try to point out both the promise and some of the technical glitches that we are struggling with along the way. Just a couple of disclosures. I should let you know that I'm a scientific founder and an equity holder in a spin-out company from the University of Washington called Cytocardia, whose focus is cardiac regeneration. I also would like to say that our company has no money whatsoever. So keep it in mind, please. Maybe the most important thing I should do at the beginning is say that along the course of this talk, if you hear me say I mean we. If you hear me say we, I mean they. This is very much a team-based effort. I don't know what exactly I do anymore. I mostly seem to sit in the office, go to a lot of meetings, talk on the phone, that kind of stuff. And these are the folks up here who do the work that I'm proud to get up and represent. So let's start, just give you a quick sense of the problem that we're interested in addressing. Heart disease has emerged 10, 15 years ago as the #1 cause of death in the world. It's replaced infectious diseases and so forth that are on the wane, and unfortunately cardiovascular disease is waxing worldwide. It's the #1 cause of death in the United States. It's the #1 cause of hospital admissions, and of course then that makes it the #1 healthcare expense, and this is the thing that's really driving the burgeoning healthcare costs and #1 Medicare expense and all that sort of thing as well. So it is the single biggest one. It's a classic chronic disease, and we'd like to see if we can do something about this. The pathophysiology is shown here. So I'm a pathologist, as Eirini mentioned, and it starts with coronary artery disease. There's actually great programs here at Sinai in vascular imaging and atherosclerosis and so forth. But typically a coronary atherosclerotic plaque grows. This plaque ruptures. There's a thrombotic event that occludes the coronary artery. This obstructs the flow to the myocardium downstream. Now, the heart's got a super high metabolic rate. It's one of the most metabolically active tissues in the body. And you get about 15 minutes with no blood flow before cardiomyocytes start to die. And then this wavefront moves from the inner wall of the heart toward the outer. This march, it's about 80% done in 3 hours, and by 6 hours, say, There's the wavefront is essentially marched through the wall and the infarct is completely evolved. That means reperfusion doesn't limit the amount of damage. So there's great percutaneous coronary interventions, thrombolytic drugs, that sort of thing. So people have made great progress in stopping the progress of the infarct. The rub has been that patients wait too long to seek medical attention. So the concept of door-to-balloon time, when somebody comes into the emergency department with a myocardial infarction, this has been essentially Toyotified. It's very streamlined. We're very efficient at this. But in the 25 years or so that I've been paying attention to onset of chest pain to coming to seek medical attention, this has not budged one iota. So until somebody at Sinai isolates the denial gene, I think it's going to be, we're going to be dealing with this for a while. So the heart is unfortunately about the least regenerative organ in the human body. The heart and the central nervous system are probably the two least regenerative. And so what happens is after myocardial infarction, there's no cardiac regeneration. Essentially, the entire infarct heals by forming scar tissue. The scar, of course, is non-contractile, and this leaves patients with a contractile deficit. And this is not a steady state. It's not like just a square wave drop with a flat plateau. This is a disease of rundown. And so there's sort of this inexorable progression in far too many patients. Patients spiral down in cardiac decompensation and develop heart failure. Once a patient is diagnosed with heart failure, it's quite deadly. On average, one has about 5 years to live from the time of diagnosis, which makes it more grave than most cancers. And so our idea is pretty conceptually simple. This is a classic disease of cellular deficiency. We can generate essentially unlimited quantities of human cardiomyocytes in the laboratory. Can we just take stem cells and use this to repopulate the wall of the heart? That's the notion. 20 years ago, when we set out on this, I figured this would be about 5 years to the clinic, and now I think it's about 2 years to the clinic. So you can do the arithmetic and extrapolate for yourself and see where this is going to go. So let me outline, give you a quick 15-year history in one slide just to bring you up to speed. My students hate it when I do this because each bullet point is like 4 PhD theses. There's been a lot of work done in trying to repair the heart with adult stem cells and adult somatic cells, cells that really aren't very stemmy at all. Excuse me, the principal sources have been from bone marrow, from the heart itself, and from adipose tissue. The basic lessons from these is that the adult cells do not engraft in the heart. And of course, if they don't even engraft, they can't go on to form new cardiomyocytes. And so they act principally through what we're now calling paracrine mechanisms, secreted factors that influence the inflammatory response, the vascularization, the fibrosis, and things like that happen. These can have salutary effects on wound healing, but they aren't bricks and mortar remuscularization of the wall. When these have gone into clinical trials, they have had very limited efficacy, and most meta-analyses seem to think that there's really not much impact yet. Pluripotent stem cells are more complicated. They're lagging behind in terms of their progress to the clinic, but it's quite clear that these cells can form definitive cardiomyocytes. They'll beat in culture. I'll show you this in a moment. And they engraft long-term in the heart if you do the experiments properly. One can form new myocardium in all kinds of species, mice, rats, guinea pigs, and so forth. And they work, we think, by a different mechanism of action. They form new myocardium that will beat in synchrony with the surrounding heart muscle, and we think that generates new force-developing units. Of course, they could be working through paracrine mechanisms as well. And clinical trials are just beginning on this. Some work in Paris, work coming in Japan, and we hope work coming in Seattle in a couple of years. So let me just give you a quick highlight. We decided to move into non-human primates as a test system because I thought this would be the best possible model of human disease. I wanted to see what we could do to step up the pace of discovery and get ourselves ready to take a shot on goal. Conversely, if this was not going to work, we wanted to fail as quickly as possible. So we went to non-human primates, and this is work led by a very talented postdoc in the lab, James Chong, who's now an assistant professor at the University of Sydney. So what did we learn from our first, what we now think of as a pilot study? It took us a four-year pilot study. One of the encouraging things is if you put in a whole bunch of cells, and we put in about 750 million cells, you can get a lot of new myocardium out. So mass in, mass out. And so there was robust engraftment. And as I'll show you, these grafts coupled electrically to the surrounding myocardium of the host, and they would beat in synchrony. They mature. That was very important. They're very immature cells, essentially fetal stage cells when they differentiate from pluripotency, but they will mature in situ in response to whatever factors the adult heart provides. They become vascularized by the host coronary circulation, and one of the big surprises is they cause ventricular arrhythmias for a couple of weeks after we transplant them. We didn't see that in mice, we didn't see it in rats, we didn't see it in guinea pigs. We went into the monkey where the heart rate was slow enough and this ventricular tachycardia emerged. I'll talk about that today as well. So the questions that our next study, the next 4-year study, wanted to address was, well, this is great, it looks wonderful under the microscope, but does this have an impact on the mechanical function of the heart, number one? And then number two, what's the mechanism of these ventricular arrhythmias? And the notion is, of course, if one can understand it mechanistically, then we should be able to have a rational plan of attack. So just a couple of quick methodological points for how we did this efficacy study. We needed to do big infarcts so that we would have a contractile deficit that would be so we could readily measure an improvement based on therapy. So we included the left anterior descending coronary artery in these macaque monkeys for 3 hours. I'll show you what the histology looks like in a moment. We had to learn how to do monkey magnetic resonance imaging, and we did this all in a blinded fashion so that the people who read the scans were unaware of which treatment groups animals belonged to. We again used large amounts of cardiomyocytes. These came from human embryonic stem cells from the H7 line, originally derived in Wisconsin by Jamie Thompson's group. And we grew them up in large vats and then froze them back and then thawed them and delivered them straight into the heart. We differentiated them. I'll show you this with using small molecule-based Wnt modulation. And this was surgical delivery. This is not how we do it in patients, but catheter delivery in a primate is not practical. The heart's too small. We tracked the animals electrically using telemetry so that we had 24/7 analysis of their heart rhythm, and the terminal endpoints were at 1 and 3 months. Shown on the bottom here is an example of what the histology from one of these hearts look like. The green is healthy myocardium, the red is scar tissue, and you can see there's a big chunky myocardial infarction here in the left ventricle, essentially transmural, and these infarcts averaged about 25% of the left ventricle. We did function by magnetic resonance imaging scanning, and what you can see, this is the left atrium, left ventricle is down here, and you can see the aortic outflow tract. I'm a little reluctant to show this here because there's great cardiac MRI that's done here at Sinai. But if there are aficionados in the audience, you may see that the heart's contracting pretty well at the base, and the apical region is hypokinetic. It's not squeezing very well, and that's because it's an apical infarct. That's the distribution of the artery that we occluded. And so this causes a big hit to contractile function. Oh, and I should make mention that this was a relay race of really good postdocs and staff who conducted this study. So Yenwen Liu, who's now a faculty member in Taiwan, Billy Chen, who's in an interventional cardiology fellowship at the moment, and Zhulan Yang, who's a permanent staff member within my laboratory. We have upped our game in terms of the way we manufacture cells. We started out doing traditional cell culture in 2 dimensions, and when you want to scale, you do what's called scaling out, which is essentially working towards a football field worth of surface area that you have to have in order to grow cells. It's quite impractical if you really want to go towards a clinical trial. So we've gone to stirred tank bioreactors. I'll show you one in just a moment. And we've developed good manufacturing practices that are so we can make essentially pharmaceutical grade human cardiomyocytes. This is in our cell and gene therapy laboratory in the University of Washington Medical Center. And everybody gets to wear spacesuits and that sort of thing. The manufacturing works using small molecule-based Wnt modulation. We basically take cells when they're pluripotent. We activate the Wnt signaling pathway with a small molecule. This induces mesoderm formation, and then we block the Wnt pathway using another small molecule, and this directs the mesoderm cells to form cardiomyocytes. This is really quite robust. It seems very simple. It's like anything. Once you reduce it to practice, it's quite simple. It took us 5-6 years to learn how to do this, but now it works quite well. This is a stirred tank bioreactor. So we grow little spheroids of cardiomyocytes like you see over here. And they, in fact, I'll just start the movie. And these little spheroids, if you squint, you can see they're beating a little bit. And these ones, we did a genome editing technique where we put a calcium indicator protein into them. It's called GCAMP. It's a variant of green fluorescent protein. So you can see them flash green every time they have a calcium transient. We did this so that when we transplanted the cells, we could do calcium imaging and have our cells report back to us on their biological activity. So we could see if they were alive, if they were beating, if they were in sync one with another, and then if the phase of their contraction was in phase with the contraction of the host heart. So that's what you're going to see here. This is a black and white movie of calcium imaging. You're looking at about the bottom 25% of the primate heart. So this is the apical view of the heart, just from an external epifluorescence microscope. This white streaky stuff turns out to be the fluorescent human myocardial graft. So this is the infarct region, and there's a good chunk of this wall that's been remuscularized. And if you do this epi-illumination, you can actually see the tissue shining through in terms of its calcium transients. So if I can get the movie. Let's see. It's a little hard to see, but focus on this spot right up here. I'll get the pointer out of the way, and I think you can start to see that there's cyclic fluorescence there. And once you see that, you can train your eye to see it over here, down here, over here, that sort of thing. It's all flashing, and it's flashing in synchrony. So what that tells you is that the grafts they're alive and they're beating in sync one with another. And that's what you're seeing here when we do offline image analysis. You can see there's 3 different regions of interest, 3 different graft regions, and their calcium transients, the fluorescence therefrom, is all in synchrony one with another. So that's cool. What your eye doesn't tell you is what is going on with the host heart, and that's shown here when you overlay the electrocardiogram, and you can see each QRS complex of the EKG is what is triggering the beat of these human cardiomyocytes. And so this was a breakthrough moment for us, because what it told us was that not only had we structurally remuscularized the wall of the heart, the new myocardium was beating in synchrony, and this was an absolutely essential feature that we had to have if we were on our way towards regenerative type healing of the infarct. So the bottom line of this study really was to see if we had an impact on left ventricular function. And I'm pleased to say that yes, we do. We have a very positive impact. So again, each line here represents one non-human primate. These, again, I should mention these are pigtail macaques, Macaque and Nemestrina. And at pre-infarction, these animals have ejection fractions about like healthy humans. And for people who are not cardiovascularly inclined, the ejection fraction is the amount of blood that is squeezed from the left ventricular chamber with each beat. And it should be over 50% if you are a healthy human or a healthy monkey. So you can see all of these monkeys are sort of in the 60 to 75% range. There's good overlap between the groups. Red is treated and blue is control. After infarction, prior to treatment, you can see there's been a 20-plus point drop in ejection fraction. All the animals really show quite consistent declines in their left ventricular function. The groups separate out after treatment, and so you can see there's minimal change in ejection fraction in the animals that received a vehicle control, whereas all the animals that received our human cardiomyocytes showed significant improvement 1 month later. Another way that you can look at that is to use the change in ejection fraction, so the difference between the 4-week value and the baseline value. And what you can see is there's about a 3% improvement in ejection fraction in the controls. That's probably within the error of measurement of MRI. And we have about an 11% improvement in the animals that received our human cardiomyocytes. So the difference between these two is 8 ejection fraction points. So you could say 8 points. Is that a big deal? Is that a little deal? It's 8 points. I can say that if you compare it to what the current standards of care are for acute myocardial infarction, it's better than what reperfusion therapy gives, it's better than what angiotensin-converting enzyme inhibitors give, and it's better than what beta blockers do. So 8 points, I submit if we could do that in people, that'd be a pretty big deal. But that was just 4 weeks after transplantation. Maybe they weren't done. Maybe we interrupted a process that was still ongoing. So we took a couple of animals out further. These are the data that I just showed you. It's scrunched a little horizontally, but same data set that you saw before. And then there are 3 animals, 2 treated and 1 control, that we followed out for an additional 2 months, so a total of 3 months of follow-up time. These are really tough experiments to do. These animals are heavily chemically immunosuppressed. And the medical management of them is quite tricky. So that's why you're only seeing this in a subset. But you can see these guys are representative of the whole, significant drop in ejection fraction, about an 8-point improvement in the animals that received our cardios, minimal improvement in the animal that did not. What we were quite gratified to see was that when we took these out for an additional 2 months, as you'd expect, the controls show no significant improvement. That's the natural history of disease. But we saw substantial improvements in both of the animals that received the human cardiomyocytes, essentially back up to normal range, maybe a 90% or so recovery of the lost function. So we would be hard-pressed if we just looked at those scans to know that there was a myocardial infarction simply based on ejection fraction metrics if we didn't know that these were part of a study that had this in its protocol. So this is the thing that gets me out of bed in the morning. It's the thing that makes me, if we could do something like this in patients, we would make a very large impact on this enormous public health problem. That's what gets me out of bed. This is what keeps me up at night. This problem of ventricular arrhythmias. And I don't want to try to minimize this or sweep it under the rug or say it's not a big deal, it's just a little VTAC, something like that. It's something that we want to understand and get a handle on. Stereotypically, this is how it plays out. The animals start out in sinus rhythm. Macaque has a heart rate of about 120 beats a minute. A few days after we put the cells in, we start to see irregular beats. Here we have non-sustained ventricular tachycardia, which typically goes into fairly significant stretches of sustained ventricular tachycardia. It starts to slow down into this curious rhythm that cardiologists call accelerated idioventricular rhythm. I think of this as basically slow VTAC. I think these are the same rhythms, just one, it gradually starts to slow down. And then they come out of it. Typically, the animals, after a couple of weeks, will come, they go into the woods and they come back out, I think, as these grafts mature. But what's going on during this vulnerable period? Colleagues who were versed in clinical cardiology said, Chuck, you've got to go and map these using these electrical mapping catheters. And okay, where am I going to find somebody who knows how to cath and map a 10-kilogram primate? And my chief of cardiology, Rob McClellan, just looks at me like I'm a fool and says, Chuck, we have people who cath 10-kilogram primates all the time. They're called pediatric cardiologists. And I was like, oh, yeah, great. So I connected with this really good pediatric cardiologist named Steve Sessler, and Steve led this series of electrical mapping studies. So we put this catheter system from Biosense Webster in. This was an earlier version of the catheter. It's a unipolar electrode, and we touch it on all sorts of places in the left ventricular surface. And you end up creating a map. And just to give you a reference, this is a kind of a funky-looking anatomical map. These three balls here would represent the cusps of the aortic valve. So that's the base of the heart. Down here in the lower right is the apex of the heart. And this region up here is where you can't get the catheter, so that's unmapped myocardium. And then you relate this to the timing of electrical activation to the surface electrocardiogram, and you can see where the electrical activation breaks out. All mammalian hearts should activate at the apex electrically, and depolarization should spread up towards the base because that's where the aortic outflow is for the left ventricle. When we brought this animal in ventricular tachycardia, and its heart did not activate at the apex like it should have. It was activating here in the mid-wall in where I think is the anterior septum. Let me just play a little movie to give you a better sense of this. So you can see it activates here in the mid-wall and spreads out both towards the apex and towards the base, which is not the way the heart is supposed to activate. We were wondering what kind of an arrhythmia this is. The arrhythmias are broadly divided into two general categories. There's disorders of slow conduction that create these zones of reentry where an impulse goes around and around and self-excites, kind of like people do in the wave in the stadium. Or you can have disorders of impulse generation, and you can think of this like an ectopic pacemaker where it's just going ping, 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 ping. Oftentimes, if it's a zone of reentry, you can actually find a rotor that goes around and around. You can actually map this and see it. We could not see anything that looked like that within the resolution of our system, which is not great. It's a clinical mapping tool. All we saw was a point source. We did a series of other interventions on this. We paced the hearts And we could take an arrhythmia that was going at 220 beats a minute. We could capture it and take it up to 300 beats a minute. But when we shut the pacer off, it went immediately back to 220 beats a minute. We did not really overdrive suppress. So if this is typically reentry arrhythmia, you can capture it and it will go back to sinus rhythm after that. What about shocking the heart? So we tried DC cardioversion. We shocked the heart, a brief pause, and then straight back to ventricular tachycardia at the same pace. So that had no beneficial impact. Typically with reentrant arrhythmias, one can interrupt the rotor and you go back to sinus rhythm after that. The last thing that we tried was what's called programmed stimulation, where we put an electrode towards the base of the heart and deliver a rapid chain of impulses that typically kick the heart into ventricular tachycardia if it is prone to reentrant mechanisms. These were no more inducible than control hearts were, so we couldn't reproduce this by pacing algorithms. And so this is clinical evidence. It's not like the sort of genetic level evidence that I'm used to, but our evidence based on, our conclusion based on clinical evidence was that this was not a disorder of slow conduction and reentry, that this instead was a problem of impulse generation that I think of really as ectopic pacemaking. I think our cells are acting like little pacemakers and are sending out electrical impulses at too high a rate that capture the heart. Let's look at some of the structural things and then we can wind up. This is a control infarct, a low magnification fluorescence micrograph. It's a chunk of the wall of the heart. You can see this 5mm scale bar down here. This is the non-ischemic vascular bed stained with a troponin antibody that recognizes both human and monkey myocardium. This is collagen stained in blue. You can see the wall is thinned. It's a chunky transmural infarct. We also stained this with a human cardiac troponin antibody, but there's not any human graft in this. And so the wall is thinned, and it's a fairly nasty transmural infarct. This is what the what the treated infarcts look like. This is a cardiomyocyte engrafted heart. And again, the same scale bar here. You can see there is a blue infarct region, but this is replete with new myocardium. This is human myocardium that's been identified with this human-specific epitope that we found an antibody to in human cardiac troponin I. And you can see a tract of maybe a centimeter or so of human myocardium that's been generated. And then it's both in the infarct region and in the peri-infarct region. This is pretty much where we target our injections, and our injections are just with needle and syringes delivered by primates like me. And so I don't think these are terribly migratory cells. I think they basically bloom where they are planted. But I used to, when we were getting going at this, I would show super high magnification images, just a few little cells, and they'd be bright green and they'd have sarcomeres and they're wonderful. But I'd say, oh, look, we're regenerating the heart. But now we've progressed to the point where we can actually show you like a whole slide scan. It's the sort of thing you can hold it up and see it with your naked eye, that sort of thing. What kind of cells are they? Everybody knows that there's at least 3 flavors of cardiomyocytes. There's atrial cells, ventricular cells, and pacemaker-type cells. It turns out that we're lucky, and the protocol that we're using to differentiate gives us mostly ventricular cells. We're using some markers where for the regulatory light chains of myosin. So myosin light chain 2V is the ventricular-specific isoform, so red is ventricular, and then green is a promiscuous one called myosin light chain 2A. So that stains atrial cells, pacemaking cells, and immature ventricular cells. You're looking at the graft-host interface right here. The monkey myocardium is down below, and of course these are all ventricular cells, and so they're red. This is the graft region here. This is 1 month after transplantation. They're mostly red with some green. That means they're mostly definitively ventricular cells, but there are some green cells that are like maybes. If we look at the animals from 3 months, it's almost entirely red, where we find, we focused on a green cell in the center so you'd have something to look at, but it's really almost entirely myosin light chain 2V, the ventricular isoform. If we quantify this where each column is 1 monkey, you can see it's about 88% definitive ventricular cells at 1 month, and it's 99% definitive ventricular cells at 3 months. And so what I think this is time-dependent maturation, and they undergo this myofibrillar isoform switch that's characteristic of the ventricular myocyte in a time-dependent manner. Just one other thing to show you about maturation. They do another myofibrillar isoform switch that's characteristic. People have demonstrated that as you go from a slower fetal heart to a faster adult, at least in the mouse, it goes this direction. They flip from slow skeletal troponin I to cardiac troponin I. Turns out human cells do the same isoform switch as well. And so the immature isoform is going to be in green. The mature isoform is going to be in red. And this is what the grafts look like at higher magnification at 1 month. It's a little hard to tell that it's muscle. You can see some nice myofibrils with sarcomeres here. But there's a lot of green and there's not too much red. At 3 months, it flips. There's a lot of red, not too much green. So you can see there's upregulation of cardiac troponin I, the mature isoform. And this looks much more like proper myocardium. So the cells are getting chunky and hypertrophic. And maybe that's why we're getting improvement in myocardial function between 1 and 3 months, is because the cells are getting bigger and stronger. At least it's true. Whether they're related or not, I can't prove. The last thing that I'll show you here with regard to maturation is that they form these structures called T-tubules, which are invaginations of the sarcolemma that allow for synchronous contraction of the heart. And so these green things that you see here are caveolin 3 stained T-tubules running down deep into the cytoplasm of the cardiomyocytes. And again, that's a sign of an adult cell. And this is just fun. Some of the grafts are big enough that we can see them by magnetic resonance imaging. There's a technique that you can use to image the infarct called delayed gadolinium enhancement. Gadolinium is an MRI bright contrast reagent, and so it lights up the infarct because it marks all the extracellular space. So here is the same heart at 3 different times at baseline post-infarction. This is the back wall that excludes gadolinium. Here is the infarct with the red dotted line around it, and it's bright because there's a lot of extracellular space. 1 month after transplantation, there are these 2 dark blobs that are appearing in the infarct that weren't there before, and they persist 3 months afterwards. Our MRI experts said, Chuck, I think what we're seeing here is new myocardium. And I said, boy, this better be pretty obvious by histology if we're going to try to claim this. And so this is a slide that corresponds approximately to the region in the red box, stained again with a cardiac troponin I that's human-specific. And you can see, again, there's about 1 centimeter or more of new human myocardium that's present. So I think again, and here's a centimeter scale in the MRI scan. So I think we're growing enough new myocardium that we can actually see it by MRI scanning. So what have I told you today? That you can take human cardiomyocytes that we generate from pluripotent stem cells and use those to extensively remuscularize a myocardial infarct in macaque monkeys. And we don't grow teratomas, we just grow new cardiac muscle. This improves global function and regional function at 4 weeks, and I think we get further improvements at 3 months based on the limited set of animals that I showed you. These are mostly ventricular cells. And we get this transient arrhythmia problem that we are working on, and that at least we know something about the mechanism now. I think it's ectopic pacemaking. And what I'd like to leave you with is the notion that, there's no sensible reason I can think of why human cardiomyocytes should be better for monkeys than they would be for people. And so I think this has real promise as a human therapeutic. What do we have to do to make this work? We've got to find a way to deliver our cells without having to open somebody's chest. I think we can do these with catheters now. We've got that working pretty well. We need to get around the issues of the immune system, so we, like many, are working on universal donor approaches. And we have to control this issue of engraftment arrhythmias. So with that, I'll stop. I'll just say thanks to the many people who made this possible. I won't go through all of this. I want to thank the taxpayers of the United States and many foundations and things that helped pay for a lot of this. This was a big one, y'all, and we're very grateful for the support that we got. And again, it's a whole team with our primate center and all that sort of thing. Couldn't have done any of this on my own. And with that, I'll stop, and thank you very much for your attention.